Good afternoon, everyone, and welcome to the Van Andel Institute's virtual public lecture series. Our apologies for some technical difficulties as we try to get started today, but we're grateful that you were willing to join us and walk through that stuff with us. Today's public lecture is on a focus on metabolism and influence on future generations. My name is Brett Holloman. I'm the Chief Philanthropy Officer at the Institute, and we're grateful that you're with us today from wherever that might be, your home, your car, your office, whatever. Today's public lecture is actually very timely. In the United States, September is actually recognized as National Childhood Obesity Awareness Month. According to the Centers for Disease Control and Prevention, nearly 20% of children in this country have diabetes, making children at higher risk for other chronic health conditions such as diseases such as asthma, sleep apnea, bone and joint problems, and type 2 diabetes. On top of this, the CDC says that children with obesity are also more likely to suffer from social isolation, depression, and lower self-esteem, all of which play into a person's overall health and well-being. Today's presentation brings to the forefront VAI's expertise and research that may have far-reaching implications, not only for those at risk for obesity, but also for all of us who wonder how our actions today may affect the lives of our descendants. We're joined today by Dr. Heidi Limpratel and Dr. Andrew Pospisil, two Van Andel Institute scientists and experts in the areas of epigenetics and metabolism. Heidi and Andrew will provide insights into growing evidence that suggests that what we eat, how we live our day-to-day lives, and environmental factors that we're exposed to may affect the health and disease risk of our children, grandchildren, and maybe even beyond. Our first presenter today will be Dr. Heidi Limpratel. Heidi is an assistant professor in the Metabolic and Nutritional Programming Group at VAI's Center for Cancer and Cell Biology. She investigates how the dietary choices of parents may impact the health of their offspring. She hopes to translate her findings into new ways to prevent disease and create a healthier future. Our second presenter, Dr. Pospisil, joined Van Andel Institute in 2018 as the director for VAI's Center for Epigenetics. In his research, Andrew seeks to understand the underpinnings of genetics and how our susceptibility to diseases may be defined from the very early on in life. One of Andrew's long-term goals is determining ways to predict lifelong health outlooks at the time of birth or even before. You will have an opportunity to ask questions through the chat feature. So as we move through the program today, feel free to engage there, and we'll have a chance. We'll hear from Heidi first, then Andrew, and then we'll wrap up with some question and answers at the end. Please join me in welcoming Dr. Andrew Pospisil and Dr. Heidi Limpratel.
Good afternoon, everyone, and thank you for being with us today. In today's lecture, Dr. Pospisil and I will try to give you an overview of what researchers in the Metabolism and Nutrition program at the Van Andel Institute study and how your metabolism impacts you and your health, and maybe even the health of your children. The Van Andel Institute's major research areas are skeletal disease, Parkinson's disease, and cancer. And almost exactly 2 years ago, a new focus on metabolism was established. 4 of us moved here from Canada and Germany, and we were joined by 2 excellent researchers who are already at the Van Andel Institute. I put metabolism here in the middle because it touches all of these diseases in some form. Cancer cells, for example, have a very different energy and nutrient demand than other cells, and scientists hope that this can be leveraged for future treatments. It has also been shown that people with metabolic syndrome, which is a group of conditions, including high waist circumference, high blood glucose, for example, and high blood pressure, they have a 24% higher risk of developing Parkinson's disease. So, okay, let's start. So what is metabolism? Metabolism is the sum of all of these chemical reactions which contribute to everything that's going on in our bodies. They break down nutrients and provide building blocks for dividing cells. They make us grow, they make our heart beat, they make our brain work, and when something goes wrong, this can have detrimental effects. Without metabolism, there would be no life. So, but don't worry, we're not gonna be having a lesson on all of these chemical reactions that you can see on the screen here. We'll zoom out a bit. And Here you can see that we like to think of metabolism at different levels. So we like to think of it as the whole body metabolism. It's the food we take in, and it gets broken down into different nutrients. And these influence very much our well-being, our weight, and everything. These nutrients and metabolites then travel to organs and tissues in our body. And they have very different demands, energy demands, for example. And just as an example, our brain, which weighs only about 2% of our body, uses up about 20% of the glucose and 20% of the oxygen in our body. Importantly, organs are made out of different cells and cell types. So take the brain, for example, again. A neuron in the brain will have very different energy needs than a supporting glial cell. So let's start at this very small level. As I mentioned, each cell in our body has a different demand for nutrients and metabolites. So every cell has its own cellular metabolism, which depends on the availability of nutrients and metabolites in its surroundings, but it also depends on the identity of the cell. Like I mentioned before, a neuron in our brain has very different energy demands than one of the supporting glial cells. Astrocytes, for example, which are a type of glial cells, take up glucose and they convert it into lactate. This lactate, however, leaves the astrocytes and it's shuttled to the neurons, which depend 100% almost on this lactate as their primary energy source. So you can imagine that if anything goes wrong with the astrocytes, it's the neurons that suffer too. In our program, Doctors Ning Wu and Connie Krawczyk study metabolism mostly at this very cellular level. Dr. Wu studies the interface between cellular metabolism and cell communication, and Dr. Krawczyk, on the other hand, studies the metabolism of specific types of immune cells and how their energy demand changes during infection. If we zoom out a little bit, and now we look at the organ tissue level, each organ in our body plays a different role when it comes to metabolism. Some organs are directly involved in metabolism, like for example, the liver or fat cells. And some other tissues do not play an active role, but they're big energy consumers, like the brain, like I mentioned before. Dr. Brian Haab and Russell Jones focus their research on what's going on at the tissue level. Dr. Haab studies pancreatic cancer. The pancreas is the only insulin-producing organ in our body, and it's essential for the control of our blood glucose levels, our blood sugar levels. Pancreatic cancer is one of the most deadly ones because it gets recognized really late, and Dr. Haab is working on developing a diagnostic test to detect pancreatic cancer at early stages. Dr. Jones studies metabolism in the tumor environment, and he investigates the relationship between tumor cells and immune cells and how they compete for metabolites. And he wants to find a way to weaken the cancer and strengthen the immune system through metabolism. Before we go into details of what Andrew and I study in the labs, I would like to introduce a few key concepts which might be unfamiliar to you. Both me and Dr. Pospisil mainly focus on obesity, which is a metabolic disorder leading to excess fat storage. Obesity, however, is not a new disease. And what you can see here on this picture is a statue which was found not very far from my hometown in Austria. And this little lady is about 30,000 years old. And historians think that it represents a symbol of fertility. So for a long time, overweight was associated with wealth and health because most of the population was struggling with undernourishment most of the time. Today, unfortunately, we are struggling with the opposite. Most of the world population is suffering from overnutrition and not from undernutrition. And the forecast is that more than half of today's children will be obese by the age of 35. You might ask, what's so bad about obesity? Unfortunately, obesity brings along with it many medical complications and comorbidities. Certain cancers, like pancreatic cancer, for example, are more prevalent in groups in people with obesity. But also gynecological complications, for example, like infertility, are increased. Here you can see maps of the US, and this one here shows obesity trends. And then all of the other ones are comorbidities, and you can see how well those maps overlap. Here you can, for example, see stroke and how it overlaps, or colon and rectum cancers, which are associated, which are a lot more prevalent in the obese population. Another problem is that we have witnessed a tremendous surge in obesity in the last 45 years. And this surge in numbers is most well documented by the Center of Disease Control and Prevention. And you just saw this little video of this rise that we saw between 1985 and 2010. 35 years, however, is not long enough to attribute these changes to genetics, because 35 years is about one generation time. So people reason that this trend has epigenetic underpinnings. But let's slow down a little bit here and define the terms genetics and epigenetics. So your genetic information, which is also called your genome, is encoded in DNA. And this DNA is like a blueprint. It is essentially the same in every single cell of your body. So your skin cell has the same genome as a liver cell. A genetic unit is called a gene, and each gene encodes for a specific protein, which in turn is involved in determining a trait. So genetics is the study of how traits, such as eye color or hair color, or the risk of developing a disease, for example, breast cancer, are inherited in very specific patterns from parents to their children. Depending on our genetics, our genotype, we display different traits and characteristics, which are called our phenotype. So now let's define epigenetics. Epi actually means above in Greek, and so it means above genetics. Your DNA determines a lot of your traits, such as eye color, like I said before, but a lot of traits are much more complex and are determined by many, many genes. In such complex networks, every player can be fine-tuned, leading to a slightly different outcome. This fine-tuning is carried out by epigenetic processes, which are involved in the packaging of the genome. Human DNA, if you would unroll it, is about 6 feet long, but it has to be rolled up and packed into a cell nucleus, which is 0.0002 inches in diameter. So some of this DNA is packaged very tightly, and some of the DNA is packaged a little, slightly less tight. And this results into different outputs from individual genes that are in this particular stretch of DNA. In the end, the type of packaging determines which genes are expressed. So this means which genes are producing proteins and how highly these are expressed. And this is different in every cell of your body. So every cell type has its own way of packaging its DNA, and this gives the cell its identity. The very fine tuning can be influenced by our environment, such as our level of fitness, our nutrition. And this tuning can go wrong sometimes too and lead to diseases without any changes in the DNA itself. I would like to provide one analogy, how I like to think about epigenetics. To me, it's a bit like music. So here I chose Antonio Vivaldi's La Primavera. And Vivaldi composed the music, and every musician relies on his composition. However, every musician has their own way of interpreting this music. And I'll play 2 examples to illustrate this to you. First, we'll hear a version of by Voices of Music, San Francisco-based ensemble with Alana Yousefian as the soloist. now I'll play the second version, a different version. And this is by Black Violin. It's an African-American duo from Florida. And it's it's two classically trained string instrumentalists. So this is what epigenetics is to me. Your DNA is a piece of music, and we are one possible interpretation of this music. Importantly, epigenetics is reversible. So a change in diet or more exercise can fine-tune the traits that we are looking at. But epigenetics is also heritable, and an unhealthy, if it's voluntarily or involuntarily healthy or unhealthy lifestyle of parents around conception or during pregnancy has been shown to impact children's long-term health. And this is what I focus on in my lab. To do this in a lab, we use a model organism, which is called Drosophila. And Drosophila are just fruit flies, like the ones that are coming out now at this time of the year and sit on the fruit in our fruit bowls. You might wonder why people use fruit flies to study diseases. But about 60 to 75% of the human disease genes have a match in fruit flies. And because fruit flies live only weeks, but age as we do, aging research is really fast in flies. Flies have helped understanding cancer, Parkinson's, and Alzheimer's. They're big in dietary research. The first learning genes actually were found in flies, and they were later found to do the exact same in humans. Coffee keeps them awake. Old flies sleep less. They're ideal to study sleep. They can also get drunk and addicted to alcohol, and there's lots of studies out there that use flies to try to get to the bottom of what causes addiction. What's important for me is that flies become grandparents in 3 to 4 weeks. So they're ideal to study inheritance. And importantly, Drosophila is the insect behind 10 Nobel laureates, which is a testament to how important this model organism has been to research, to medical research. So what we do in the lab is we developed a Drosophila model for epigenetic obesity. So what we do is we expose the fathers, the male flies, to different amounts of sugar in their food. So from very low sugar, which they can hardly survive on, to very high sugar, like the sugar content of a ripe banana, for example. We then mate these flies to females that receive normal standard diet, and then we look at the offspring. So we measure their obesity. And here you can see, and this is fat levels in the flies, that if we raise these flies on normal food, there's not much of a difference. But surprisingly, when we raised these flies on a high sugar diet, so a high calorie diet, these flies, the offspring of both low and high sugar fed fathers, were more obese than the offspring of fathers that had a middle sugar diet. And so this is very similar to, I think, what we see now. If these children are exposed to normal caloric diet, they wouldn't develop obesity. But because we live in a world with just, where food is everywhere around us, and high calorie food is everywhere around us, we see more obesity. We also did many more experiments, and in the end, using these fly models, we could detect epigenetic pathways that control the expression of the genes, that control the output of metabolic genes, which were then in turn the cause for the obesity in the flies. So we showed that high sugar in the fathers, for 2 days actually only, caused a long life reprogramming and to cause obesity in the very end stage in the adult flies. We could also see a conservation of the molecular signature that we discovered in the flies in humans, which justifies the use of flies to model human diseases. So what do we do now in the lab? In the lab, we are now interested in determining or in finding out what the signal is, what is in sperm that conveys this message over to the next generation. And then how is this message then translated into a phenotype, which ends up being obesity in the end? And we are also interested in finding something that we can use to interfere either with the generation of the signal or with this translation into the phenotype. And this is to find maybe avenues of treatment in the future, to block the transition of the signal or to block the establishment of the obesity. We are also interested in a lab on the maternal side of things. So we would like to see how maternal nutrition and high sugar in particular what influence it has on the oocyte or the egg cell quality. We would like to investigate how high sugar diet and or obesity impacts this oocyte, this egg cell, and how it impacts the development of the early embryo. We are hoping to find out if obesity-related fertility problems are partly caused by the impact of body physiology on oocyte composition. We've also just recently started looking in what else has intergenerational effects. What else can do this, not just high sugar? And so we're looking into nutrients and environmental toxins, and if they have intergenerational phenotypes, if we can find something in the children of these flies. We have just recently started a screen on environmental chemicals. And so environmental chemicals are defined as those industrial compounds present all around us in our air, water, food, soil, dust, consumer products. And importantly, there's a large nationwide biomonitoring program, again by the CDC, and they were able to detect 100 out of 300 measured chemicals in people's blood and urine. And this points to significant potential health risk and puts the magnitude of exposure into context. We try to screen these chemicals that were found in the blood and urine of the general US population and look what impact the exposure of these chemicals has on phenotype of flies. And we now look at more phenotypes, not just obesity. We also look at Parkinson's phenotypes, autism, fertility, and cancer. And we will measure this measure these phenotypes in the exposed generations as well as in the next generation. Included in the screen are chemicals of specific relevance to the population here in Michigan. So one of the chemicals we will investigate is lead, and lead was the main contaminant in the Flint water crisis. Even extremely low levels of lead can cause behavior and learning problems, lower IQ, hyperactivity in children, and in pregnant women, it can cause reduced growth of the fetus and premature birth. Another chemical we are planning to investigate is the group of pair and polyfluoryl alkyl substances, short PFAS. Several sites of contamination have been found in Michigan, with one major site being just north of Grand Rapids in Rockford. PFAS is used in household products, for example, to make them non-stick like Teflon, or in grease-proof fast food containers. It is also used in waterproofing materials such as shoes, like they did up in Rockford. There's evidence that exposure to PFAS has adverse human health effects, but studies in humans are always extremely difficult, and the data are often very noisy. We hope that with our DROSOPHILA model system, where all aspects of the environment can be controlled and the diet is very defined and controlled, we can shed some light on the effects of these chemicals and the underlying biological mechanisms. Last but not least, I would like to acknowledge all the people who I've worked with at the Max Planck Institute in Germany before moving here, and who I'm working with here right now at the Van Andel Institute. As you can see, science is teamwork, and we would not be here without our collaborators, locally in Michigan or overseas, for example, in Sweden, Spain, the Ukraine, and Austria. And here, I'll pass the torch to Dr. Pospisil, who will talk to you about the research that he's interested in his lab.
Okay, thank you very much. Yeah, thanks, Heidi. So what I'm going to try to do in the next 10 or 15 minutes is put everything that Heidi said into perspective of modern medicine. And modern medicine really, today's modern medicine is drilling down towards understanding variation, trying to understand really who we are, what are the individual aspects of each of us. What I'm showing you here are portraits of many famous people. Many of them you'll know. These are from a portrait photographer. His name is Martin Schuller. He's got a studio in New York, and he's very famous for his stills just of the face. And I think what his work highlights beautifully is how unique each one of us is, right? And this really relates to what Heidi said. Heidi told us that who we are is dependent on our genes and our environment. She explained on our genetics and on what we eat, right? And I think medicine has been pushed towards this dogma because of twins. This is the cover of one of Martin Schuller's books. What it shows you are actually two identical twins, and it's half of an image of each of their phases. And this is a photo montage then. And if you look carefully, it's almost impossible to distinguish these two girls. It's very difficult to find any features. And so what does this tell us? Well, identical twins come from the same piece of DNA, right? They split in that very first cell stage of life into two individuals with the exact same copy, and then they have the same environment. So they're a test for this paradigm of, are we just genes and our environment? Here are those two girls side by side, and indeed, they're extremely similar, right? It's difficult to find differences. What you don't hear about are twin pairs like this, and they're actually very common. And those are identical twins that you can actually easily tell apart. These you might mistake just as brothers. If you look at the individual on the right, facial structure, and this is the bones, is much broader. This isn't that this individual is eaten more or less, right? Skin on the lips, shape of the lips is different. Almost every feature about these two brothers is a little bit different. So how does this fit into the puzzle? Well, if genes and environment kind of dictate where you fall on that landscape of variation of the human population, epigenetics is kind of this red range here, where it can fine tune who you become. It can push you a little bit to the left, a little bit to the right, a little bit heavier or a little bit lighter if you're talking about obesity. It also gives us insight into who else we could have been, right? It's a little bit like a roll of a dice. So just as each one of us has our DNA and environment, so did these two gentlemen. They both had the same DNA and the same environment, yet they came out a little bit differently. So the consequence there for each one of us is it kind of means that each of us could have come out differently, right? And that there's some probabilistic, that's why I show these dice here, there's a bit of a roll in the dice in who we become. And so why is this important for medicine? Well, it's important because if you do the measurements, and there's been pioneering genetic research in twins, and if you do the measurements, it turns out that in humans and in most animals, genes and environment for most traits is responsible for only about 50% of the puzzle. Now you'd say, well, identical twins are identical, so how can it be just 50%? Well, it's true. For some twin pairs, it turned out to be 100% of who they were, but for other twin pairs, it turned out to be very little. And that's that that magic that Heidi was talking about of using the DNA in different ways, right? Sometimes the two twins can use the DNA in the exact same way, so their epigenetics was the same, and other times it can be different. And that's what Heidi and I study. So how does it fit into today's medicine? Well, I think you'll know from any time you've been to the doctor's office, the traditional medicine, I would even say contemporary medicine, what we all go through every time we go to the doctor is a one-size-fits-all type of medicine. What do I mean by that? You come in, you get an examination, and if you have diabetes, you will be given one type of medication. It's not very complicated. If we have the disease, we get given the one medication. And the unfortunate side is that some of us respond very well to those medications. Others, not so well, and a rare few then exhibit adverse effects, right? And so about 15, 20 years ago, momentum got going behind a movement called personalized medicine. And that idea that with technological advances, could we figure out really exactly every individual's disease predispositions and quickly generate a therapy that's really specific and individualized to that exact person? And the key step is this step here, research and technology, and doing it rapidly enough to figure out a specific medication for a specific individual. And the benefit, of course, is then everyone could have the beneficial effect. The parts of that would be things like better prediction of disease susceptibility and movement from reacting to diseases towards predicting them and preventing them, preempting their progression, and customizing disease prevention strategies. We'd have more drugs and we'd be able to more effectively prescribe them. We would avoid predictable side effects, and we would reduce the time, cost, and the failure of pharmaceutical clinical trials. And in so doing, we get rid of a lot of trial and error that is in medicine. For some diseases, there's multiple drugs out there, and many of you may have experience with going to a doctor, they try one medication, then it doesn't really seem to work, so then they move on to their second favorite choice because it didn't seem to work. So we would eliminate those trial and error aspects of medicine that happen, and in so doing, substantially reduce healthcare costs and increase benefit. But so the latest advance in the last 10 years or so has been a transition from what I just told you, which is personalized medicine, to something called precision medicine. The idea that every single one of us requires a different therapy is a little bit far-fetched. There's 7 billion people on the planet. You know, if we all had diabetes, I don't think there would be 7 billion different types of diabetes. But there might be a few different flavors, and each of us may fall into a different category. So if we can catch those bigger, broader categories, we still may be able to get that benefit we want for all patients without the complexity and probably the unrealistic ambition of getting a truly personalized medication for every individual. So this is where we think epigenetic fits in. Epigenetics is, you know, it has been born out of wonderful observation by astute geneticists back in the day, but it's something that's also been born out of technological advances in the last 10 to 20 years of our sequencing of the genome and then being able to read out how many different flavors that genome can give rise to. I'll give you a real world example. So precision medicine is new. You won't have heard of many benefits of it outside maybe the cancer space, where in cancer you can really do that personalized approach in some rare instances where you can figure out what was the one mutation that one cancer had, and if there's a medication against it, you can try to use it. I'll give you an example from the diabetes space. Most of you will probably have this picture of diabetes in your head. A lot of us grew up learning there's two types, one that you get as a child, that's called type 1 or autoimmune diabetes, and another one that you get later in life, and it's usually associated with getting older and often with obesity. Now there's a bit of a revolution happening in the diabetes world that you may or may not have heard of. It hit the news a few years ago, but then of course quickly died down. But in the scientific space, it's, it's an exploding concept. And that is that those 2 types of diabetes have now become 5 types of diabetes, all of a sudden. And what happened? Well, some clinicians and a group of researchers in Sweden took a very large cohort of patients and just did really rigorous, unbiased analysis on them. And they found out that the patients themselves fell into 4 different clusters. So it was the type 2 diabetes patients that fell into 4 new clusters. So we still have type 1 diabetes. They gave it a new name, a complicated one. And then we have now 4 types of type 2 diabetes. One of them is because you don't make enough insulin. One of them is because you don't respond to that insulin. One of them is simply because you're getting too heavy, so it puts too much pressure on the body. And another one is simply because you're getting older. And so the system is getting a bit rusty, you could say. But the good thing for these last 2 is actually the prognosis is really good for these last 2. Their long-term stability of the disease is a slow march. Much different than these first 2, where there's a real critical problem with the system. And so what does this matter? Well, each of these groups just now, is starting to be treated a little bit differently with slightly different medications. And that's precision medicine happening in the real world. So I'll give you an example of how we're trying to tackle this in the lab and how we see these concepts play out. What I'm showing you here in these left 2 panels are the fat mass and the lean mass of a whole bunch of mice that are all genetically identical. All the gray ones are identical and all the blue ones are identical. And you can see that epigenetic concept there. If genes and environment were everything, then all these dots would be all together in one really tight cluster. But what you can see is that all the dots are different. These 2 mice over here, they're about 42 grams. They're genetically identical to this guy right here. And that one's only about 28 grams. That's a big difference, almost 50% difference in body weight, even though they're essentially identical twins. These ones we've found a mutation that exaggerates these epigenetic effects. So we've made these 2 red spots over here on the curve go further apart. And what we can do is trace where these differences come from. So what we found out is these 2 blue populations, the differences between them are because one of them has very normal insulin secreting cells that are called beta cells. And the other has what we would call super beta cells. They divide faster, they secrete more insulin, they secrete insulin better. These ones probably would have a tougher time ever getting diabetes, right? And so what brings this work back to what Heidi was talking about is we really recently noticed that this is an intergenerational effect. So just like Heidi's sugar-fed fathers, the results that you see here, this high variability in these identical organisms can be traced to the father. So on the panel in the lower right, what you see is the body weights or the fat mass of the offspring of all the different fathers that were used in this study. And you can see the fathers in the left, they give lots of the heavy offspring. The ones in the middle give kind of a mixture of offspring. And the ones on the right almost never give a heavy offspring. So something is transmitting from the father to the children, even though all these fathers, they're all identical. So this is epigenetics playing out. And so how does that relate to precision medicine? Well, you could think of these 2 groups here as 2 precision medicine groups, right? So just like in that schematic I showed you before, if epigenetics defines 2 of the groups that we'd like to, 2 of the groups of disease that exist out there, maybe we could come up with different therapies that would ideally treat each of them. And that's, brings it back to the twin analogy, right? That perhaps even though these twins are identical, even though their genetics is the same and their environment is the same, maybe we'd actually have a better treatment for the twin on the left and a better treatment for the twin on the right, that were distinct. And so as much as that applies to twins, because twins are a reflection of our own DNA template and what it could have come out as, this could also apply to each one of us, right? U1 or U2, if anybody remembers the book thing one and thing two, identical but different. So it's all about the pursuit of as precise a medicine, as personalized a medicine as possible, but without trying to get to 7 billion different versions of a medicine. So if I can bring it to the takeaways of the talk, both Heidi's and mine, I think the one thing, the three things that we'd like you to take away would be that, you know, who we become depends on genes, environment, as we've always believed, but also of some more probabilistic or variable factor that we like to call epigenetics. That the parental environment, that their diet, stresses, and pollutions can affect offspring health lifelong. And then epigenetics and understanding it is really integral to the future targeting of precision medicine to precision groups. And so I'll finish off there with my own acknowledgement slides. Mine has some pictures to show you that there's tons of young, talented people working on these problems, and each one of us scientists could never do this alone, as Heidi said. Just my own collaborations, as you can see, span many lines of text. There's generous funders, generous donors, and generous resources that are, you know, funded by the government and private enterprises. And so I think I will leave it there and pass on the torch. And thank you all for your attention.
Heidi and Andrew, thank you very much for your presentations and for challenging our thinking and changing out ways that we think about epigenetics, one, to help us better understand it, but then two, to help us understand better how it applies to precision medicine as we look to move forward. What do you consider the biggest gap in knowledge in nutritional research today? As you look out at the landscape, you've mentioned that metabolism is a kind of a newer upcoming field that, Heidi, you've said we've placed it in the center of cancer and Parkinson's and some other diseases as well, autism and others. As you look ahead, what do you see as the biggest gap of knowledge?
So I think, you know, we're just starting to understand all the aspects or all the diseases that metabolism touches on. And so I showed this slide in the beginning with all of these chemical reactions. A lot of these chemical reactions have been known for many, many, many years. And I think for a long time, we thought we know everything about metabolism because we knew these chemical reactions. I think only now do we understand how many aspects of life are controlled by metabolism. And if metabolism goes wrong, then, you know, it has detrimental effects and leads to diseases.
And Andrew, what about for you?
I think similarly, I would build on that, you know, the biggest missing pieces are, I think Heidi highlighted that complexity, are the connections, you know, which exact dietary components connect to which exact genes. We know we're learning more and more about all the different genes and what they do, but that link between the dietary components and the genes, I think that's our biggest challenge at the moment.
Yeah. As you look back on your research careers, what was kind of a moment that you thought, wow, this was a discovery I had no idea, or took you by surprise, or kind of that aha moment? What's one that stands out in your own minds for each of the things that you study? Andrew, we'll start with you.
I think I showed it. One of the images that I showed, or one of the graphs, was that that notion where all those blue dots were in two different clouds. And that really solidified for me that epigenetics is powerful enough to give us really two different and distinct and reproducible outcomes. That if you or I could have been born 100 times, it's possible that we could have come out as two different individuals, not just a range of little modifications of one, but really two different individuals. And that for me has changed everything. The analogy I love to use are insects, like the honeybee. There's the queen bee and the worker bee. They're really different. All the queens are the same. All the workers are the same. They all come from the same type of DNA. So if that's true for each of us, it really solidified for me that that precision medicine might need 2 therapies for each one of us. And we just need to figure out which one of our 2 selves we are.
Mm-hmm. Heidi, we'll turn it over to you.
Yeah, so for me, the most surprising thing was to see, and I didn't show that experiment on my slides, is to see that the parental diet, or paternal diet in this case, the high sugar that we fed to the fathers had a direct impact on this DNA packaging, like I described, right? So that when we fed high sugar to the fathers, the children of those flies had a slightly less densely packed genome in many places. So not just at the exact metabolic gene locations. And that is very hard, and we don't know how that happens. We don't know why it happens, right? But it's a very intriguing finding that we are also trying to figure out what's behind this change in DNA packaging and how this is happening just because of sugar in the father.
Yeah. For you again, Heidi, what do obese flies load different? This is a question from the audience. So do obese flies load different classes of lipid into eggs as compared with lean flies?
So that's what we like to figure out, actually. And so we have just got the first samples on our metabolomics facility here at the Institute. And that was my reasoning, right? That the metabolic load of these eggs is probably very different, but no one has actually ever looked at it. Mm-hmm. The reason is in humans, egg cells are too small. We don't get enough material out of it. And obviously it's very hard to get those. Same for mice, right? Even in mice, they're very, very small. A Drosophila egg is nice and big, and we can actually do these measurements in single eggs. So we can feed the mom different nutrients and then look in the exact eggs of that female and see what the composition is of these eggs.
So kind of along those lines, your goal or hope then, the question is there evidence in mammals that egg lipid composition changes in females with metabolic syndrome?
So we know that women, obese women or women with metabolic syndrome have infertility problems. A lot of that is attributed to the hormones because obese women produce a lot of leptin, which is a hormone, and that plays into their whole hormone balance, and that causes a lot of the infertility. It's not known, and no one has ever looked at what the egg composition means for that. And also it plays a huge role, I think, in vitro fertilization, because where the egg cells get taken out of the moms, right? And then they get incubated, fertilized, and then the embryos grow up in this solution that we think is a good composition, but we don't know because, you know, we know the embryos grow and they can be then implanted and we get babies out of them. But is that as good, or is that the best we can do, or could we improve that solution that they're growing in these very early stages? Yeah.
Along those same lines, Andrew, as you look out in research in diabetes, what do you see on the frontier of both understanding and eventually maybe treating, maybe even possibly curing something like diabetes?
I think I'd jump to sort of the outlay. I think diabetes is the pioneering precision medicine example of common diseases. So with the exception of cancer, which is many, many, many different diseases, you know, the disease that we think about most simply as a single disease that we're now learning to substructure would definitely be diabetes. And I think, you know, with increased resolution and understanding of each of the subtypes, and I'm sure in 10 years, it won't be 4 subtypes that have split off of type 2 diabetes. And in fact, there are a few more that we do know of. It'll be 10 or 15 different subtypes. And I think what that allows is not just precision medicine that we make 15 different pills for people to take, but that we get a much more nuanced understanding of why these individuals get the disease. And if we can have much more preventative solutions, such as different diets. And, you know, there's going to be plenty of the food industry companies that get in this. You can imagine, I think the easiest way I imagine it is you go to the cereal aisle at your local supermarket. There's already, 400 different boxes with different colors on them. And some of those colors are going to be dedicated towards different types of disease or people who are susceptible to different ones. And it's up to Nestle and those companies to decide how that packaging will be done, how the sales and marketing will be done. But in the end, the big leap will be if each one of us can be involved and active in preventing our own disease, our own disease onset, because we know well ahead of time what we're susceptible to.
Sure. Kind of along those lines, and Andrew or Heidi, either one, feel free to step in. Similar hope in the space of Parkinson's to be able to create, right now we consider Parkinson's disease as a single umbrella disease to some extent. As we understand epigenetics and metabolism and that sort of thing, hope for or potential to look at Parkinson's in a different way, similar to what you just articulated with diabetes?
I think for sure. And that's a fascinating, complex issue. There's already a couple subtypes. One would be already that we know is very genetic, and those are individuals that get it very early on in life. That it's almost guaranteed it's a mutation, or they're one of the different possible outcomes that we've all had. And so early onset is almost surely a genetic cause. It has slightly different etiology. For the later onset, what we're finding out, for instance, to be quite specific, is that some of it may be the result of infection. And that's something that's very active in this institute, is pursuing the notion that this may be a very slow chronic inflammatory process that was started, 20 or 30 years earlier when infection came within our gut, for instance. And neurons are very sensitive cells, and eventually they just can't cope with that syndrome anymore. So yes, if one could totally imagine that with different infections, there's going to be different varieties of Parkinson's that you get, if indeed infection is doing it. Different routes of infection, if one got it through the gut versus got it through a blood-borne infection. That may cause different disease etiologies, and then the same rules apply. If we can figure it out, if we can find a biomarker, what we call it, some representative signal that we can measure that says, okay, this individual is type A and this individual is type C, then we can design and develop medications that are more appropriate.
Mm-hmm.
I think along the same lines is that Parkinson's disease is often diagnosed when we see actually the latest symptoms, which are motor symptoms, right? Yeah. One of the first symptoms, though, for example, is losing your sense of smell, or constipation, or insomnia. And so, but every patient displays, or we don't, because we diagnose it so late, we don't know if patients display different symptoms of onset, like these symptoms that we've ignored for so long, and not just now realizing that they have been connected with that Parkinson's disease, and they have been part of it, but we only recognize the motor symptoms late on.
Yeah. So as you both look out ahead for Van Andel Institute and the Center for Metabolism and Epigenetics, what encourages you, or what do you get excited about as you look at the next 5 to potentially 10 years?
Heidi, you go.
Sure, I start. So I'm new, right? I've just established my group. So this is the first time that I lead a lab. And so to me, it's all exciting. It's exciting to be part of a new team, the metabolism program. We brought in 2 years ago, and we kind of all started at the same time. And now we see, the first successes, right? And the first, the experiments that the results that are coming out. So I'm just excited to see how each of us will succeed, and also how metabolism then goes or can branch out into these other areas at the Institute. And like Andrew said, with Parkinson's disease, there's a huge link between metabolism and Parkinson's disease, which hasn't been explored very much in the Institute just because the expertise wasn't around. But now that we are here, you know, I think we're all excited on getting involved in the other aspects of the other diseases that are researched at the Institute.
Sure. Andrew?
For me, I think the exciting, I mean, it applies to the Institute, but maybe to science as a whole now is just technological advance and the pace at which things are changing for us as a scientist. And I mean, what we've been enabled to do by technology. When I think just to my PhD, you know, a few decades ago, what we were able to do at that time is nothing compared to what we can do now.
Mm-hmm.
As a scientist, that's a bit stressful. As you get older, it's tough to keep up with the young whippersnappers and technologies. You know, every day there's a new paper with a new technology, and you know, some reviewer will tackle you and say, well, why aren't you using the best, the newest technology? So that can be stressful, but at the same time, we get to see through the graduate school here at the Institute, you know, a young new generation that is used to growing up in this fast-paced world, and they can handle balancing and always having an ever-changing landscape of information, and they learn to leverage it a little bit better than the rest of us. So that's what excites me, is in 10 or 20 years, I'm sure science will be moving at a pace and with a power that I can't imagine right now, because certainly at the time of my PhD, I would never have imagined some of the things we can do now, and that we can do quickly. Within a day, we can do stuff that would be unimaginable in a lifetime back then.
Sure, it goes back to the slides that both of you shared in terms of the important role that collaborations play and the complexity that it's not just the work that you do alone, but the support that you have within your laboratories here, as well as your collaborations abroad. Well, this brings us to the end of today's lecture series, and we're thankful, Heidi and Andrew, for your willingness to educate us, to share, to give us some things to think about, for us to understand differently. For those of you in our audience, you can stay connected with us at Van Andel Institute at www.vai.org. You'll find upcoming events located there, additional lecture series, and we just thank you once again for taking the time out of your day to join us and your dedication to understand science and the role that it plays in the health of humanity as we look ahead. I think in our current time, we see the role that science plays as we better understand COVID-19 and how that impacts human health and the diseases then that Heidi and Andrew, you study. So thank you so much for bringing that to us. Our work continues. It looks a little differently these days. We don't have everybody in the lab all day, all the time, but certainly we press on to understand the diseases that we can impact human health in a positive manner as years and decades unfold. So we hope to see you around in the future, whether it's a virtual event or whether it's an in-person.
No.
We'll wrap things up. My computer is muting me, so I think that's the sign that we need to sign off. We're thankful for your participation and grateful for your engagement with Van Andel Institute. We'll hope to see you at an event sometime, either live or in person or again virtual. Check out the website, and thanks so much for your continued engagement. Have a great afternoon.